Let's begin. Today I'm going to be talking about a few things. Before I share my screen, let me look at my grade book and state that I've graded a number of things in the class. And I think it was over the weekend. I don't remember when I did quiz 1A and quiz 1B and quiz 2B. But quiz 1A is finished. I need to do quiz 2A, and hopefully I'll get that done at least by the weekend. And I'll work on it this week, but I'm not going to guarantee I'll get it done by this week. Because quiz 2A, I got to read the questions and then read your answer and then decide on the grade. And that takes a long time to grade that quiz. But I've got a large number of the labs graded for lab 00, lab 1, lab 2, lab 3. I got that graded the second time, meaning I got it graded the first time weeks ago, but I got them all graded this weekend for the second time. So you can take a look at your grade there and what my comments are to your answers. I've started grading lab 5 the first time, and I've graded lab 4 the first time. So take a look at those. And I think, let me look at this. I think you can make corrections for full credit by Saturday, but let me make certain that's true. That's true for lab 5. It looks like lab 4 is already shut. Let me look at that further. Yeah, I've got the last date to do lab 4 as the 16th. So this, maybe I got it graded before the weekend. Let me take a look at that. Oh, let's see, how do I do that? Let's get out of here. Go to the speed grader. Yeah, I got that graded October 12th. So this was done earlier than this weekend, lab 4. But for corrections, you can do lab 5, although I've only got about half of them graded. And I also graded the lecture worksheet to the last chapter. Was that chapter 4? Yes. All right, any question about any of that? I do need to grade the extra credit quiz 1, and you can take a look at what your answers are for the multiple choice questions. And I've got to do the grading for the essay questions. I'll try and get towards that this weekend as well. Any questions about any of that? All right, let me get out of the grade book and now I show my screen. We are recording. Yep, okay. So today, if you look at what we're doing, we're not doing a lab. I'm going to discuss the unknown project, and hopefully I will mail you your unknown number tonight, although it might be late tonight, but I'll try and get that out tonight. And then you will start the unknown project tonight if I succeed in getting that out. And initially, I'm going to be telling you which tests you're going to do. And the purpose for this is that I figured out that with just one class, this project will take over 100 emails for this one project. And I decided that that was too many emails to do for one project. So I'm assigning you 3 lab tests that you're going to be doing. You don't get any choice. You have to do these 3. And I will just email it to the entire class. And so instead of sending out 60 emails, I'm going to send out 1 email. And that will greatly reduce the number of emails for this project. Any question about that? So this unknown project is normally in the lab, you would come into the lab and you would be given an unknown bacteria, and then you would streak it out for colony, getting a pure colony and then growing up a pure culture, and then running different tests on it to try and identify the bacteria. Well, we're an online class, so obviously that can't happen. And what I'm going to do is I'm going to give you an unknown number that's going to be representing your unknown bacteria. And you will be running different tests on this unknown number, the unknown bacteria, to try and identify the bacteria. And when you request a test, that test has to incubate, and you can take a look. Let's go to the lab. Under week 5 lab, it shows you the different tests that you can run. It's called week 5 lab unknown test. All of these different tests are tests you can run to help determine what bacteria you have. And once you request a test, let's say you request the biolisculin test, you can click on this and get what the test is and how long that test needs to incubate. So when you send me an email, actually, you're not going to send me an email. We'll do it in the unknown notes page, and we'll discuss that in a minute. When you request a test in the unknown notes page, that test will then incubate for the time that that test requires the bacteria to incubate. And you can click on it and find it out. For the biolisculin, that test has to incubate for 2 days. After 2 days' time of incubation, I will then give you the test results for biolisculin. Now, what I'm going to do is send you pictures. And then you're going to look at the pictures and make your observations, and then write down your observations. That might be like what color it is for biolisculin. And then you make an interpretation. Your interpretation for biolisculin will be, is it positive for biolisculin or is it negative for biolisculin? The observation will be the color, and biolisculin can be really dark brown or black color, or it can be the normal color of the tube. And I don't remember if I've got a picture on that one, but you can get pictures online, or yeah, there is a picture on this one. So that would be a negative, that's a negative control, that's a negative bacteria. And this is a positive, one where the tube turns black. Any question about that? That's your observation. The interpretation is for this species, biolisculin negative, for this species, biolisculin positive. You write that down in your unknown notes, and then you look at the table for your results and the possible bacteria, narrow down the possible bacteria, and then you ask for another test. And that's how this procedure will be done. It'll be all done online. And you won't be handling any bacteria, but I will be giving you results, pictures for your bacteria. And then at the very end, you'll try and identify what bacteria it is. How you can determine what bacteria it is, look at the unknown list. Let me open that. And there's something like 40 different species, maybe 50 different species. Let me open it in this, it'll be better. This table has the different tests on the top and the different species down the bottom. Hopefully that'll open here. There we go. So these are the possible species. These are the tests you can run to identify your species. Any question about that? Now, I will be determining the first 3 tests you're going to run. You're going to be doing gelatin hydrolysis, the gram stain, and PR lactose. Okay. Any question about that? After the first 3 tests, you will decide which tests to run. So that'll be totally up to you. And I'm going to have you set up the gelatin test immediately. It takes a week to run. The gram stain, you're going to be setting it up in about 5 days time, and you'll get the results in a week's time. And the PR lactose you'll set up in PR lactose you're set up in 6 days from now, and you'll be getting the results in about a week, because this test needs 1 day to run. And that way I can send all 3 of the results in one email. Normally you'll ask for 1 test like the PR sucrose. It will then have to incubate, and it incubates for either 1 day or 2 days. And I will then send you the results, and then you'll see, okay, it's AG positive. And let's say that PR lactose was AG positive. And so that narrowed it down to these 4 species. And then you'll run a test to try and narrow down these 4 species. Any question about any of that? So everything will be done online, and you will do, everything will be done except for my first email. Everything will be done in the, what's it called? I've got it here. The unknown notes page. Everything will be done in the unknown notes page. So you ask for tests in the unknown notes page, and you will put your observations in the unknown notes page. You put your conclusions in the unknown notes page, and then you write anything else like what you're thinking about and why you're doing a different test. Okay, and it tells you what to do in the unknown notes page. So after the first or 2 emails from me, record your daily activities, and especially your test requests here. I will then provide the results here. What you're going to be doing is every time you update this page, you're going to have a button here for uploading a document. So upload a document. You can put an infinite number of documents on your unknown notes page. I, on the other hand, I don't think we have any student who has anything. I, on the other hand, will contact you in the comment section. I don't want you guys putting comments in here. That's for me to give you your results and for me to give you any necessary comments. If you put a comment in there, I'm going to delete it and tell you to submit it as a document. And I'm an instructor, so I don't have the link here, but I believe you guys have a link up here for submitting the document. Any question about that?
I have one. How long does this unknown project or assignment, is it supposed to like go from the time that you send us the email until the last day of the quarter that it's due, or?
It's the last day of the quarter that it's due. It's near the end of the quarter, but it's not the end of the quarter. Let me look at the schedule here, because I don't remember the exact date. The unknown project report is due on Saturday, December 4th at 11:59 p.m. Okay, so that's the week before finals week.
Okay, but then it starts as soon as you send us this email, like like it's,
Yeah, it'll start as soon as I send you the email. And like I said, when I send you the first email, you're only going to get the unknown number. You need to put the unknown number in your unknown notes page, because when you ask for a test, if there's no unknown number there, I can't give you the results. So you need to put your unknown number in your unknown notes page. But like I said, I will email you the first 3 tests, and I told you what those 3 tests are. You need to put the results in the unknown notes page, because that won't be in this page. And so everything is going to be in the unknown notes page. And then both you as well as I can look at the unknown notes to determine what you've done. Okay, so everything has to be on your unknown notes page. And this actually works very well. Do not send me a test request in the email. If you do, I will tell you I'm not accepting test requests in email. So you have to put it in your unknown notes page. And the reason is because I want everything together. And so if there's ever a question, everybody can go into the unknown notes page and look it up. Any question about that part?
So we just message you tonight our number and the request for those 3 tests?
No, you don't need to request the 3 tests, because I'm telling you, you have to do those 3 tests. Okay, so you can put in your unknown notes page, once I send you the number, that you were running the gelatin hydrolysis test the night I send you the email. And you will start if I send it today, you will start the gram stain 5 days from today, and you'll start the PR lactose 6 days from today, assuming I send you the number tonight. Okay, so it'll be 5 days from whenever I send you the email. So that can go in your unknown notes page. And then when I send you the email, which will be about a week from when I send you the first email, I will send you the results to those results to those tests. And you do need to put those results in your unknown notes page. And you should put your conclusions in your unknown notes page. Okay? A result is an observation. A conclusion is your interpretation. So the color of the tube is an observation. Whether the tube is positive or negative is an interpretation. Most of the tests will have a positive or negative outcome, but there are a few, like the PR glucose and all of the PR sugars. There is a negative, but there's no positive. There's either A or AG. Okay. And like for the hemolysis, well, there is a negative on that one too, but it's actually called gamma. Let me see where that is. Why am I not seeing it? There it is. Scroll down to gram positives. So that one will be alpha or negative or beta for beta hemolysis, alpha hemolysis, or negative for no hemolysis. Okay, so the table gives you the results for the different species, and it also tells you about some of the tests and how to read it. Let me bring that over here. So down here, it tells you how to do the gram stain. Let me blow this up because I can't read that. Poor eyesight, I've told you that before.
Aren't you going to be giving us the results? And
I'll give you the results, but you got to interpret it. Meaning I'm not going to give you the results. You're going to get a picture and then you have to even state what your results are. Okay?
yeah.
Okay, so for the gram stain, to make a gram stain from a broth, it tells you how to do it here, although you should do the thing. For the cellular growth arrangement, I'm going to warn you that your gram stain is going to be made from an agar stock culture, in which case the cells tend to stick together because they're growing in a solid agar stock culture. And it's generally best to do the cellular growth arrangement to, sorry, you can't read that here, to grow it in, actually it doesn't even state that here. I thought it did. It's generally best to do that from growing the bacteria in a liquid culture, a broth culture. And it's not stating it here. I thought it was. This version doesn't have it. So you'd have to look up, back up, cellular growth arrangement. Oh, come on.
Quick question.
Just a minute, I'm trying to finish that and then we'll get to your question.
Mm-hmm.
Oh, there it is. It's in the gram stain here. Well, assuming that's going to open. There it is. So the cellular shape and the growth arrangement. And where is that stated? It is difficult to determine the cellular growth arrangement from the gram stain. That's because we're doing the gram stain on a solid culture. So what you want to do is do this from a broth culture. Grow up your cells in a broth culture to get the cellular growth arrangement. And I'm going to change the wording of that table because that's what it's supposed to state. Maybe it states it. I'll take a look at this and change the wording of this table. All right, what's your question?
Okay. My question is, the notes, the unknown notes that we're all supposed to do, is this like a discussion thing or is this all individual? Like we all have our own notes?
Yeah, you'll have, each of you will have your own unknown notes page. There is no discussion with other students. Other students will never see your documents, files, whatever on Canvas. So this is a file for you on Canvas.
Okay, perfect. And then the information that goes in our unknown notes is just the results from our tests that you run?
It states here what you're supposed to put in your unknown notes page. You need to put in your observations for the results, the interpretations, obviously you put in what test you're running, and then you should also state everything you do concerning the unknown project will be recorded here. Okay, so you should be talking about why you're running a certain test. You should be putting in what your bacteria species could possibly be, and you're running this test to narrow it down. Okay. And like I say, make it a submission document. Don't put it in the comments. All right. Any other questions on the unknown notes page or the unknown project in general?
Yeah, how many tests are we gonna be running other than the 3 that you're gonna give us?
It will depend on each student. Some students run as many as 10 tests. Other students, the luckiest student may only run 4.
How do we be that lucky?
You just have to be lucky.
Is it the same answer? Like, are we all guessing for the same?
No, you'll each have a different species, if that's what your question is.
That's what it was going to be.
Yeah, you'll each have a different species. All right, let me see. Let's close that down. I'll leave that open in case there's a question. I'll leave that there. All right, let's go back on that one. There we go. All right, so let's talk about the unknown project. Am I recording this? Good, I am. So this project is worth 100 points. It's a lab project, so these points will go in the lab. You will be working on it from whenever I send you the first email, which hopefully will be tonight, till close to the end of the term. You can read about the rationale. I'm not going to discuss that. I'm not going to discuss that either. The project format, you need to isolate the bacteria. Normally, we give you a tube in the lab, and it'll have 2 species of bacteria in it. We decided that that's going to be too difficult to do online. So online, I'm just going to give you a number which is going to represent 1 species of bacteria. So you don't need to do really anything to purify it, but you can streak it out for colony streaking because it's going to be a week before you get your first results. So you can streak it out. Make sure you have a pure culture. That's a good thing to do. And yes, it's an online class, so we're pretending to streak it out. Any question there? All this work is going to be online. So you're going to be pretending to work with a pure culture. So you're going to just be working with a number and then making test requests online. And then I'm going to send you a picture for the results online. Okay?
Right there on phase 1 isolation, number B, it says as a team. Do we get to have a partner or no?
If we were in the lab, you would have a partner, but we're not in the lab, so you're not going to have a partner. Okay? And even though you start as a team, remember I told you you had 2 bacteria in the tube? Well, 1 student takes 1 bacteria, the other student takes the other bacteria. So after the start, you actually work individually. Okay, but we don't do that. Like I said, you're going to get 1 number, which is going to be a pure culture to begin with. But like I said, you can streak it out or pretend to streak it out because it'll be a week before the first results come. You should look up the results and you can go to the Canvas page. In many of them, there's a picture for helping you interpret the results. You can also go online for picture. And if you have access to Berge's manual, that would be the best source. Although if you're totally online, that would be hard to do. You might be able to find a little bit on Berge's online, but certainly you're not going to get Berge's lab manual online. You need to follow dichotomous logic when you're doing this test. Dichotomous logic means you're going to run a test which can have 1 or 2 possible outcomes. Let me try and demonstrate that. Like for the gram stain, you're running the gram stain, you can have 2 results. I don't know how to draw that. So 1 is gram positive The other is gram negative. Okay, for PR lactose, well, that one's not entirely two, but for the next one, let's go over here. We have gelatin hydrolysis. There's gelatin hydrolysis. I'm gonna go G, hydrolysis. Negative. And gelatin hydrolysis positive. Okay. So if your test result is gram negative, we then end this line here, and then you go with the gram negative. And if the result is a gelatin hydrolysis, we then end this line here, and we continue running a test from this one. Is everyone following me? We started with all bacteria. We then narrowed it down to gram negatives. We then narrowed it down that those that are gram negative to those that are also gram gelatin hydrolysis positive. So you need to do dichotomous logic. And even in the first 3 tests you're running, you're doing the gelatin hydrolysis first because it takes a week to run. You're then going to do the gram stain in 5 days time. And you're going to get your results in a week's time. And then in 6 days time, you'll do the PR lactose. So in reality, even for the first 3, you're doing it using dichotomous logic. Any questions about that?
All of these instructions are written down, right? Because I think I'm super, super confused.
What are you confused on? Because no, those instructions aren't written down. I'm telling you, I'm sending you the results to the first 3 tests. And this is written down here that you have to follow dichotomous logic for this project. If you do not follow the dichotomous logic for this project, you will get a penalty on your lab report.
So for those first 3 tests, do we have to like, I just don't understand like how you said we start them today and we start one another day. Do we like go on a website or something to start them?
Remember, this is done all online. So in reality, you're requesting the gelatin today, assuming I send you all the information today. And you'll start requesting the gram stain in 5 days time. And then the PR lactose in 6 days time. And the reason is because I want to send all of this at once. And I want you to follow dichotomous logic. Okay? Meaning I want to send one email, but I want you to follow dichotomous logic. And I'm not going to be sending out 3 emails. Okay? The whole point of doing it at once, doing these 3, is to reduce the number of emails. Because 100 or more, not emails, sorry, 100 or more contacts between me and the students is just way too much for one project.
And we request all the tests on the notes page, right?
Yes,
Okay.
okay. All of your work will be done on the notes page, with the exception of the very first email from me. And then when you submit your lab report, you won't do it in the unknown notes page. There will be another file for submitting that. And let me see what the name of that's called. It's called the I don't have it here because it's not due here. Just a minute. Let me scroll down to the bottom. Why didn't that scroll? Sorry. There it is, it's called the unknown report. So you'll submit your lab report in this document here. But all of your daily work on the unknown notes should be recorded in your unknown notes page. Okay. The unknown notes page must be finished by December 3rd, and that's a day before you submit your unknown lab report. The unknown lab report is worth 75 points. Sorry, I am using an old document here. I'm pretty sure that said 100 points. Yeah, this is not correct. That's worth 75 points. That's been changed. It's been changed for a year, and I'm using this older document. Sorry about that. So this project is worth 75 points. All right. Any questions about any of that? Let's move back to week 5 lab and talk more about the unknown project. So follow dichotomous logic. Some students, every once in a while, they say, oh, well, I don't want to do that. I want to get all the results all together and get the project done with. When you use shotgun logic, you will suffer a penalty. You can, I'm not going to tell you not to, but you will get a penalty on your lab report. You should write your daily lab notes in your unknown notes page. When you turn in the lab report, which is going to be worth 75 points, it should be typewritten and hard copy, and you got to submit it in the document. Please write with 12-point font. I don't see that well, and if you make it 10-point font, I'll have a hard time reading it.
Okay, correct me if I'm wrong. The unknowns report is everything that you put in your unknowns notes, correct or no?
The unknown notes page may have more information that you would not put in the unknown labs project. For example, you might be just speculating about what your bacteria might be. You could do that in your unknown notes page. I wouldn't do that in your unknown lab report. I probably wouldn't mark you down for it, but there's no reason to do that in your unknown lab report. The unknown notes page is not formal. The lab report will be a formal lab report. However, if you write up your unknown notes very formally, there will be no difference between the unknown notes when you put them all together and your unknown lab report. So those of you who write formally, the unknown notes will be the same as or very similar to your lab report.
Do you have an example of a lab report that we can look at so we know what we're expected to put in it?
See that sample journal right there? There's an example. I don't want to talk about that yet because I'm still giving you the instructions. Let's go back to the instructions. Oops, I hit the wrong button there. Something's opening. Oh, Outlook, let me close that. You should not omit data that makes your project look better. And since everything will be in your unknown notes page, I can look at it and make sure you're actually running the test you said you ran and whatnot, like in the correct order and stuff like that. Your unknown notes page will be dated automatically by the system. However, in your lab report, when you're talking about a test, you should put in the date. Any question about that? You may add drawings, photographs, and tables. They are very helpful, but they're not required. There is one table that is required, and I'll talk about that in a little bit. That'll be near the end of your lab report. Each report entry in your lab report must have a date and a time. It's not so important to have the time for an online class. Most of you can just put the date. Very few of you will be requesting 2 tests in one day. But if you do request 2 tests in one day, you should have the date and the time. In the lab, if you were to actually come into the lab, it's important to have the date and the time because some people could come into like the 11:30 lab, and then they might come into the 5:30 lab and run a different test. And so you need to have both the date and the time. But online, that's most of you will be only doing 1 test per day, in which case you only need the day. If you do more than 1 test per day, then add the time. You do not need to describe the step-by-step details for each test you run. All you need to do is say, I ran the gram stain. I know the steps of the gram stain. You do not need to tell me the steps of the gram stain. The thoughts and words of your report should be your own. Remember, if you do not use your own words, you have to put it in quotes and then state who you got it from. Otherwise, it's plagiarism. It is mandatory that your lab report have a table outlining the process you use to identify your unknown organism. There is a sample table in this report as well in the sample example of a lab report. So here's a table. First, the student here did a gram stain. And before they did this gram stain, it was all the species and they didn't list them. After the gram stain, it was these species. They then did the catalase test and it was catalase positive. And so that narrowed it down to 4 species. They did the PR mannitol and it's PR mannitol negative. That narrowed it down to 3 species. They did the PR glucose. The result was acid. That narrowed it down to 2 species. And then they did the citrate test and it was citrate positive, and that narrowed it down to 1 species. So you should give a table or a diagram showing me how you came up with the species that you think you have. Any questions about any of that? If you run a test which doesn't give you any information, meaning no information, you don't need to add that to the table here. Like let's say you did the, I don't know which would be a good one. We'll say PR lactose test and it gave you the same 4 as the catalase test. You don't need to put that in the table because it doesn't help you identify your unknown species. But all other tests that help you identify your unknown should be included in this table. This table is required as a part of your lab report. If you don't have the table, you'll be missing something and you'll get a deduction. Any question about that? Where were we? At the end of the lab report, you should have a paragraph, and it should not be longer than a page. It can be as small as a paragraph, describing the organism that you think you've identified. You should give a literature citation for where you got that information. And if you ever got information elsewhere in your lab report, that should be in the literature cited page. So the very last part of your lab report should be a literature citation. And please use a correct citation style. I don't care which one you use, although be consistent. If you're using APA citation style, make sure you always use APA citation style. I got a warning about plagiarism. The grading. Let's talk a little bit about the grading. The test has to be performed using dichotomous logic. I'll be grading you on the completeness of the report, what tests were ran, why they were ran, possible conclusions, what were the results, what are the conclusions. Remember, a result is an observation, a conclusion is your interpretation of that observation. Diagrams, photographs, and tables can be helpful, but they're not required, except for the last table, which shows me how you identified your unknown. And it can be either a table or a flow chart, meaning a diagram. You'll be graded on organization, clarity, and neatness. This part is done for those who go into a lab, and you guys are not doing the lab, so we won't actually do a test where we're streaking out our unknown on TSA, azide, and McConkey's. And then this is giving you an example of a dichotomous process, meaning using dichotomous logic. If you're having trouble with dichotomous logic, you can work through figure B, an example of dichotomous logic. There's some questions here. You do not need to answer these questions for the project. This is just to help you with dichotomous logic if you're confused on what is dichotomous logic. Okay? All right. Let's take a look at what your lab report will look like. There's a sample right here. The sample lab report was actually done by an instructor many years ago. And I probably should open that in a new page. Come on, open. It's downloading, just give it a minute. There it is. Oh, no, it isn't. It's right here. So this has something I don't want to see, and that is they have a cover page. Put all of this information on the first page of your document. Any questions about that? You should state each daily entry with a date, and if needed, a time. So this stated I streaked out 2 TSA and 2 E and B plates. We're not going to do any streaking, although you can streak out to purify your culture from day 1 because you're not going to get the results for a week. So let's skip this because, like I said, this is something you're not going to be doing on the first couple of days because they're streaking it out. And as you can see, he had a culture of 2 different bacteria. You're not going to get a culture of 2 bacteria. My mouse is dying for some reason. There's yellow colonies and white colonies. So let's go down here. And this is the gram stain. So he did the gram stain and he says it looks like it's gram positive cocci. And as you can see, these look like a staphylococci, those look like a streptococci, and that looks like a diplococci. So do not take the cellular growth arrangement on the gram stain. And then he stated that because I had gram positive cocci, I wanted to run the catalase test. So he's talking about what activities he does on a day, meaning that would be the test you're requesting. You should write down your observations. On this day, he had no observations. And then the conclusions from the catalase test was he was catalase positive. You don't have to have observation, activities, and conclusions. Most people probably will do activities when they request it, and that will be a different day than their observation. And then they'll give their observation, their conclusions together. Either way is fine. Okay, you can have your observations first. And what he's doing is he's giving everything on one day that he's doing. On this day, no observations. He decided to do the catalase test, so that's an activity. And then the conclusions to the catalase test. On the next day, no observations, because when he did the test, he got the results on one day. And then he stated he wanted to do the hydrogen peroxide test. So that's catalase. Why is he doing the catalase again? I don't know. Anyways, bubbling, catalase positive, catalase negative. Let's go down to another one. He did the mannitol broth test. He requested that. In the previous day. So what day is that? February 11th, 2004. And then he got the results to that test, February 12th, 2004. And the mannitol broth test was an A, an acid former. And then he's got a little table, which you can do, it's not a bad thing, what his results have been. Gram-positive, the shape is coccus, the pigment is yellow, the catalase is positive, hemolysis is beta hemolysis, bile-esculin negative, and then mannitol, that's PR mannitol, positive. The possible species, he wrote down Staphylococcus aureus. All right, any questions about the example? Everyone good?
Yeah.
Okay, I'm going to assume everybody's good. All right, what more do I need to talk about? I need to talk about the grading. There's the report grading rubric down here. You will be graded on the utilization, you'll be graded on the utilization of resources. If you use excess media or you run an irrelevant test, you won't get full credit. None, you get full credit. So that one will be, let's see if this is right. Yep, total 75. So that is out of 10. Were the correct species included in each step? And let me go to the example. He's only got one here, so let's back up. Here, in this day, he had two possible species or maybe two different family. Let me see what that is. Staphylococcus or micrococcus. So he narrowed it down to a family. You should state your species, just not just the family. But that will be okay. All right, here he's got Micrococcus luteus, but it's not beta hemolytic. Staphylococcus epidermidis is not yellow, so it's not, and it's not beta hemolytic. So those have been removed. So the possible species of 3 at this point are only Staphylococcus aureus. My point is at each step, you should be stating what species you have, and you will be graded on that. There is that. There it is. No, it isn't. Here it is. The completeness of the report. If all the data is presented, you'll get 10 points. If there's one instance of missing info, you'll get 8 points. 2 to 3 missing info, you'll get 6 points, etc. Effective and clear communication. If there's no grammar, spelling, or punctuation error, you get 15 points. If there's a few errors, but logic is understood, you'll get 13 points. If there's lots of errors and it's hard to read the paper, you'll only get 1 point on that. The correct species is identified, you'll get 10 points. If the species identified is within the same group, same type, you'll get 7 points. If the species is outside the gram stain, meaning you say, I think it's Micrococcus luteus, which is gram positive, and I tell you, no, it's E. coli, which is gram negative, you'll get 5 points on that. And so it's a total of worth 75 points. Any question about that part? So when you're writing up your lab report, you might want to take a look at the how it's going to be graded. At this point in time, you don't need to know how it's going to be graded. All right, are there any questions? I've got a link for the instructions right here, which I've gone over. How to build a dichotomous game plan is right here. The different species under consideration is right here. And this also gives the tests that you can run, although it's better once you've figured out a test to come down here and take a look at the test in the unknown test, because this will tell you about the test. It'll also give you how long the test has to incubate. The longest test is the gelatin hydrolysis, and it takes a week to incubate. That's why you're doing that one first. The shortest is that you will get the results on the same day. And I think catalase, there's only two, catalase and that's the other one. I don't remember. Turns blue, oxidase, catalase and oxidase are the only two tests which you may get the results on the day you ask for it. All the others take at least 1 day. Most of them take 2 days to incubate, and one of them takes 5 days to incubate. So if you ask for the MR test, you're not going to get the results for 5 days. All right. There's a sample example. And there's the link to the unknown notes. I've got a link to the unknown notes at the bottom of the lab. So like week 10 or something, as well as, this is week 11, as well as your unknown lab report. So you'll submit the unknown lab report here in your unknown notes there. Any questions? If there's no questions, that's it. So I will see you on Thursday.